Well, thank you for the invitation. I really appreciate the opportunity to tell you about some work we've been doing in our lab. Had some great introductory lectures about SERS and using it for diagnostics. Duncan even talked a little bit about imaging. I'm going to combine a little bit of that, and we're going to talk about SERS imaging in live cells today. Maybe even allude to some things that might help address some of those questions we had earlier about location of reporter molecules in cells. That's where we're going. All right, so let's get started here. When we talk about cellular imaging, most of us are used to seeing these kinds of images, which are common. This one's actually stolen from the Nikon microscope objective guide. It was on the cover a few years back. And what we're looking at when we see this is not necessarily the cell, but we're seeing stains that have been attached. And this relates back to the beautiful molecules that Duncan showed us earlier that he's using with SRS. And in this case, we have something yellow that stains the nucleus and something magenta that hits the mitochondria, and those cytoskeletal filaments are this blue-green. And all of this is dictated then by preferred interactions for those parts of the cell. And what my lab is interested in is trying to understand what's in the cell without having to use a stain or an affinity reagent if we can help it. And there's some cases you can't. And so the question is, how can we do that? And the simple answer is we can use Raman. And so we've had some really nice introductions to Raman imaging. What I'll show you here on the right is what happens if we map this Raman band over here at 1660-ish in these prostate cancer cells. If I hit play on this, you can watch, and as we move through it, we can actually see where this tween nanoparticle drug delivery agent localizes throughout the cell. I'll run it again so you can kind of see it. You can kind of see nice localization. And this is just spontaneous Raman. In this case, we're taking advantage of the fact that the tween forms a micelle. And so the local concentration is actually higher than if it was homogeneously dispersed through the cell. And we can see it down into micromolar type levels. So that works okay because we have this higher local concentration, similar to what people have seen with lipid droplets and nonlinear imaging. It's this spontaneous Raman. It takes a little longer than the SRS that Duncan's talked about, but you can use the vibrational spectrum to map out things. In this study, what we also did is we looked at a nutraceutical lycopene, which is found in tomatoes. It gives it its red color, but it's also been shown to inhibit prostate cancer. And we were able to actually map out where in the cell the lycopene seemed to go. And in particular, shown here on the left, so this is the dark field image of the cell. And now this is the lycopene image, which is extracted using the vibrational modes of lycopene that are detected within the cell. Again, just using spontaneous Raman. And so where these are, we can see it doesn't seem to go to the nucleus. And we've been able to show that with some of that volumetric imaging that I showed in the last slide. But we see that it does go inside the cell and seems to localize around organelles that also scatter in the cell. And so we're able to use, the spectrum then to specify, where the lycopene is, and also say a little bit about things like cis-trans isomerization that occurs in this molecule as it's being metabolized. In the case of lycopene, we can actually get down to fairly low limits of detection, micromolar, but that's because it's resonance enhanced. So there's an electronic transition near 532 for this long, conjugated chain that increases the Raman signal. And most molecules that we're interested in don't have this. And so the question has been, how do we do Raman imaging, of naturally occurring molecules when those things have much, much lower signals? And that's really where the SERS comes into this for us. And so we had a really nice, the plenary lecture about space. And so, a little bit coincidental here. And I've used the Mars rover kind of as my analogy for what the nanoparticles are going to do here. We can't see what's going on the surface of Mars basically from here, but we can send the Mars rover to go and sample that. So what we've done in our lab is we've used nanoparticles as a way to put them into cells to sample the local environment, increase the scattering of molecules that wouldn't otherwise be detectable, and let us see that back in our Raman images. And so we've had enough people talking about SERS, I don't want to belinger the point, but this plasmon resonance basically increases the scattering from molecules near the surface, such that with certain molecules, you can go down to a single molecule type level. For many biologically relevant molecules, you can get down to biologically relevant concentrations using the full spectrum again to resolve what you're looking at. My lab's been doing this for some time now, and one of the systems that we're preferential to is this one here, which is the RGD integrin binding system, looking at how integrins, which are extracellular matrix proteins, receptors on the outside of your cell that bind to the extracellular matrix, can be regulated and targeted using, plasmonic nanoparticles. This is a nice system to study because it's been shown that arginine, glycine, aspartic acid peptides will mimic the fibronectin fibers in the extracellular matrix and let us use this to target our nanoparticles to these protein receptors as a model for protein ligand binding. And so we can put our peptides, our RGD peptides, on the nanoparticles, and then put these on cells and then image where the receptors actually are. This has been interesting work. Using the spectrum that you get out, you can say something about how specific or selective a receptor is for a certain peptide. And so we've looked at various isoforms of that RGD receptor. So the green would be the RGDFC that I showed you kind of on the last slide, or sorry, the blue is the RGDFC. The green is if you switch the D and to the DGRFC, you altered the peptide sequence slightly, and you can show that from the spectra that you get a highly conserved spectrum in that case. If you get rid of the phenylalanine, what we see is that the spectrum changes quite dramatically, and so you start to see interactions with other parts of the cell. I should point out that most of the features that we detect over here on the right, which are representative of those clusters of spectra, don't come from the RGD peptide on the nanoparticle, but are actually characteristic of the protein they're interacting with within the cell. So our probe is feeding back information about which protein it's actually interacting with. And you can do things like watch how these particles diffuse and bind in live cells and see how changes in diffusion coefficient actually correlate to the Raman specificity that we can get out using the spectrum and looking at things like a multivariate analysis plot of that. So, a long time ago in a galaxy far, far away, so they might say, we started doing this experiment where we just had proteins on surfaces, and we saw that if we bound nanoparticles to our proteins and then did this technique called tip-enhanced Raman, which is really, really elegant and really, really hard, we could get those signals out. And we showed that you could even translate that to cells. This is not high throughput. In our more recent efforts, currently, we've shifted away from using tip-enhanced Raman and using some of these more elegant shapes like nanostars that Kimberly just referred to, due to their higher enhancements, to be able to get the same kind of information out of cells, but without having to do this fancy tip-enhanced stuff. And this has enabled some really cool things, including the ability to look at live cells using these plasmonic nanoparticles. So, how do we do that? It's basically the same strategy. Oops, sorry, I went to a slide too far. We take our RGDFC peptide, and now we just decorate it using the cysteine gold chemistry to decorate it onto these nanostars that then we can incubate with our cells and culture, and then we can even keep them alive while we're imaging to see where the nanoparticles have bound to detect that spectral information that I was referring to in previous slides. There are a few trade-offs with SERS imaging over the TERS imaging. With TERS, you had really high spatial resolution on the order of 10 to 50 nanometers, but you had a limited scanning area. With this SERS mapping now, we can actually do whole cells in the same amount of time as this little area we could do with TERS. The resolution, the spatial resolution is now diffraction limited, but we're still getting out this wealth of chemical information talking about what the peptide on the nanoparticle is doing as far as targeting to the receptor on the cell. We've been able even to develop this into like a SERS specific diagnostic or assay for specificity, right? So we can take the spectrum that we know corresponds to the receptor of interest, and then we can do like a Pearson's correlation against the spectrum observed at every point in the cell. So this map over here is kind of fun. What the little numbers inside the red boxes is the Pearson's correlation coefficient to the known spectrum of a peptide receptor interaction. And so we can score all the places that we detect Raman signal to determine the average correlation across a field of view or a cell, and then use that to say what the specificity of that peptide functionalized nanoparticle is to that specific receptor. And so for the RGDFC nanostars, we see a much higher binding, a higher correlation coefficient than if you do the same experiment with just bare nanostars that don't have any peptide on them. Conversely, if you were to block the RGD receptors just using RGD peptide in solution and then try to do this, you also don't see any nanostars binding, again, if we unblock it and do our probe cells, then we can see a really high degree of specificity with high statistical significance. The p-value here shown is 0.004 for the blocked versus the unblocked, and 0.008 for the two types of nanostars. All right. What's been cool about this is because we get the spectrum, we can start to look at what amino acids seem to be involved in this type of binding interactions on the cell surface. And for the integrin receptor, so this is a model taken out of this paper of the alpha V beta 3 integrin binding to this RGD peptide. And we can see, what the protein structure predicts for the amino acids nearby, and we can compare that to the signal we get and what functional groups are detected in the SERS spectrum. And we see there's this tryptophan W129 that's pretty close to the binding site of the protein. And that really seems to be what's driving the signal that we see. This is an agreement with some previous work that we reported back in this Faraday discussion in 2015 that shows that the tryptophan residues in proteins are just much, much brighter than the other amino acids as far as their SERS signals go. And so having a tryptophan residue there can help give you a brighter signal that you can see quite easily in these SERS maps. We've done some other work I'm not going to talk about today explaining that, but it does give a very, very bright, probably a surface enhanced resonance Raman type response in these interactions. Where I'd like to go next is talking about how we can use those interactions to actually improve upon the spatial resolution in this wide field SERS imaging. And this has led us to do what we call super resolution SERS imaging of these protein receptors. And so the idea is the same nanostar binding, we can now do a wide field image where here we're just bending all the photons to see where things are lighting up, but we can apply super resolution techniques then to localize that back down to a much finer spatial resolution, similar to what we had in the TERS experiments to begin with. So if you're not familiar with super resolution methodologies, the way this works is you illuminate the whole cell at one time. So this is the red laser being dispersed across the entire cell. And we take the picture with the red illumination. And then we take a whole bunch of pictures. And what you can do is you can fit these bright spots. And it turns out with SERS particles, they actually fluctuate in time. And so it gives you a time varying position that you can use to localize where the emission's coming from. And so in these storm algorithms, you set a threshold. And if it doesn't fluctuate within that threshold, it actually removes it as background. So this cell being applied to this two-dimensional Gaussian fit to the emitters with time that are fluctuating gives us this super resolution image. And so this big spot here resolves down to this little nanoparticle that has a full width at half max of about 60 nanometers. So we're getting back into this, higher spatial resolution imaging that we can then associate with these proteins inside cells. Just a quick note, I always forget to say this, but it's very important. We did not develop super resolution SERS imaging. Catherine Willits, who's now at Temple, was one of the pioneers for that. And she was very helpful to us when we were developing our algorithms. And there's been some other people, Alex Brolo, University of Victoria has also gotten into this game as well. Just some really exciting work here in the last few years. Coming back to our receptor imaging, so what we do is we have our nanostars now with these peptides on them that when we feed them to the cells, they bind. So this is all done in culture. And then we do imaging of those cells. We disperse them onto a cover glass and then wide field illumination, take our series of images and get the super resolved SERS image. What we can also do is we can go back to say the point here that I'm illustrating, and we can take the spectra. So we can't quite do this simultaneously in this presentation, but we can get spectra as well as images. And so this is kind of an example of this analysis. So here we have two cells, and in our movies, so like the first frame, this would be what was recorded in the wide field SERS image. And then we'd record, 1,000 frames of these very, very quickly. So in 126, we start to see this feature here light up. And so when we average all of the frames together, that's what we get here in C. And then we can take that spot that kind of lit up that showed this time variation intensity fluctuation and look at what happened in this case, over about 450, 460 frames, where we see these intense fluctuations in intensity that are actually what the storm algorithm is going to be pulling out and triggering on. And so when you apply that, you get a bunch of stuff down here that turns out to be background, which we can clean up using some other algorithms. I don't know if I have time to talk about that today or not, but we do see this nanoparticle cluster type forming. And if we look at this, the scale bar here is about 100 nanometers. So this is probably maybe 2 particles or possibly even 3 that are bound pretty close to each other. And when we take the Raman spectrum, again, we get these bands over here in the Raman spectrum that are generally characteristic of tryptophan, which suggests it's kind of the same interaction we had been seeing in our prior work with nanostars and just doing point imaging as we went through cells. If we take that particle and one of these particles that we've isolated in a cell and we look at it a little closer, what we've noticed is that we can pull out things like dynamics of the protein receptor on the cell membrane. So this was the localization of one of those particles, those nanostars on the cell membrane, over 1,000 frames. And if we look at this, this spot is about 60 nanometers full width half maximum, which is what I told you earlier. If you look at the time evolution of the localization, where the emission is coming from on the cell, what you notice is that there's a pattern. So the early times are the dark spots and the late times are the yellow spots. And you can kind of see there's like this oscillatory motion in the localization. If we take that and we spread it out in 2D and time on this axis here, what we see is the particle seems to be wagging back and forth. So initially it kind of went over here to the left, and then it came back to the right, to the left, to the right, just kind of stayed put, then it started wagging again. And the localization precision here is about 6 nanometers. So we seem to be able to monitor how the plasmonic nanostar is kind of moving attached to this receptor as it's bound to the surface of the cell with very, very high spatial precision. If you look at in the bottom row here, this is what happens without the peptide on the nanostar. And in fact, the localization is nowhere near as close. This is more or less diffraction limited, slightly improved, because you would expect the emission to come from random points on the star, whereas in the case with the peptide, the emission actually comes from the protein bound to that RGD ligand in one spot, which increases the localization. And then again, without the peptide, you don't see any of this, correlated oscillatory motion occurring in the cells the same way. And you can do things like measure the diffusion coefficients of the 2 probes based on these types of measurements, and you get the same kind of biophysical information you would get out of single particle tracking that has been used in other implementations to study this. So what we've been curious about then is, can we study this, to use this to study where things are occurring, not only on the surface of a cell, which is important when you're doing things like TERS and studying these extracellular membrane receptors, but can we also go inside the cell? And so because our nanoparticle doesn't have to be on the outside of the cell to be detected anymore, we can start to section through the cell. And here we've gone in 1 micron slices through about 5 nanometers, and we can show that the particle really only shows up and localizes in the same focal plane of view. And so in these 4 different focal planes, we found these 4 different particles where we were able to section through the cell, kind of a micron at a time, to see where the particle was in that cell. All right, so this is all really cool, but it really kind of frustrated me that we were losing all of our spectral information while we were getting this super resolution localization. And so we started thinking a little bit about other things we could do. And so one of the things we did was look at pH sensing, and Duncan gave some really nice probes. I'd actually like to play with his probes at some point too, to see if we can do some other sensing. But MBA, mercaptobenzoic acid, shows really nice spectral changes with pH. And so we started making these pH sensitive probes as a way to test, can we see spectral differences as these nanoparticles are taken up into cells, largely associated with the idea that endosomes have a different pH than, say, the cytosol, than the extracellular space, and being able to image that. And so we have these probes that show a nice pH-dependent change. And there's even this frequency shift here at about 1380 intensity change that we might be able to resolve and to help to tease out what's going on. And we can do this in our point imaging and detect the spectrum, such as what's shown here in this example, without too much problems. We're taking this now into super resolution. You're coming on to tell me I'm running out of time, right?
2 minutes, yeah.
All right, I think I can get there in 2 minutes.
Perfect.
What we realized was that we could resolve both the super resolution position along with the spectrum at the same time if we just limited our field of view. And so what we do is we take our image and we kind of cut it off to about a 35 to 40 micron region, and then we pass it through a diffraction grating before recording it on the sensor. And so the diffraction grating splits our light up into the 0th and the 1st order, at least that's the parts we're catching on the sensor, where the 0th order diffraction gives us our image of our nanoparticles, and the 1st order gives us the spectrum of the same particles simultaneously. And so we can take a single frame here, and you can see a bunch of particles, and you can see these streaks that are associated with particles. You can average it to increase your signal to noise. But because we're taking these in single frames, we can also apply the storm fitting to localize, in this region where you can't even tell because, the spots are so small relative to the contrast that there are these 4 particles that gave rise to spectrum simultaneously. And so we're able to go back and get the spectrum and the position at the same time. Doing this, we can actually resolve dynamics on, tens of hundreds of milliseconds, looking at things that are changing around these particles. And we've been able to do some DFT calculations to kind of model that. And we can show that the fluctuations seem to arise from changes in the molecules. I'm running out of time, so I'm going to skip over this a little bit. One of the cool things is that if you localize where the fluctuations are occurring, you can see with this really high precision again, that the fluctuations in this case, which we associated with formation of an electron transfer radical, seem to occur at the ends of these octopod type nanostars, where the fields are predicted to be strongest, and then the closed shell species, the MBA molecule on the surface, seems to correlate in the center. We've been able to make SERS tags out of these molecules now and actually embed it with our cells. And so now we can actually do this live cell imaging where I've cropped the regions just to show the 0 and the 1. Here's where it's in focus again, and we can get the spectrum of MBA. It's not quite as high spectral resolution as what I showed you from the point imaging, but it does give us the peaks that are characteristic. The intense peaks can clearly be seen over the scattering background of the cell. And it's also highly sensitive to the focal plane still too. So just like I showed you, we could do sectioning with a super resolution. We can also use the spectrum to section because you really only see the intensity of the peaks when you're in focus on those particles. What can you do? It turns out there's one last thing I wanted to mention. It turns out while we're super resolving the position, we can also super resolve the spectrum, so to say. And so it turns out because we're fitting the spectral fluctuations the same as we're fitting the position of the emitter, we can pull out an improved peak position from the storm fit on the spectral domain of our images. And so while our spectral resolution, from the limited by diffraction on the grating and the size of our slit is only about 20 to 30 wave numbers, we can kind of pull out the centers from the storm fitting of where the fluctuations occurring with much higher precision. So here, this is a case where there was really no change in the nanoparticle, and we can see those 2 spectra. When we start to see the fluctuations, we can pull out where those centers are occurring and where the fluctuations occur, giving us improved chemical information of what's happening around these nanoparticles, even while they're inside of cells. And so this is really kind of an exciting way of adapting this super resolution algorithm to improve the spectroscopy of what's changing in the cells as well. All right, just to summarize, I hope I've convinced you that you can do some really nice single molecule, low level of detection spectroscopy of using nanoparticles inside cells. Detecting the spectrum really helps us validate what's interacting, what's going on inside cells, and even seeing chemistry. Tryptophan is an important thing. You can do this in live cells, and we really like this new super res spectral imaging approach. We think this is going to be a really exciting way to study some of these interactions and really understand some cell biology going forward in the future. There we go. That's what I was looking for. Sorry. I just need to thank my group. The 2 grad students who have contributed the most to the work I've talked about. Brian Scarpitti is a 4th-year grad student in my group who has been absolutely phenomenal. And he contacts you about a postdoc in the future. He's amazing. You want to hire him. And Devin Shoup is a younger grad student who started working in the group, and she's now the one pushing forward on some of the spectral super resolution imaging. A bunch of postdocs have helped us along the way. Our nanostar work was started by Sean Sloan-Denniston, who's now at Strathclyde, back working with Duncan and Karen on some other projects. And then Li Fu and Carlos are 2 former postdocs who also helped us along the way getting involved in some of this receptor imaging and super res imaging stuff. And with that, I'll stop and answer any questions that people might have.